answer to what kind of experiment can be designed and conducted. Take the lean mouse, for instance, any rodent or any other animal for that matter, and introduce high numbers of staphylococci. Keep in mind that they can also cause infection, and see if that will influence the overall status of the animal. But again, we have to keep in mind that obesity, as many other abnormalities in the status of the eukaryotic organism, is caused by multiple factors. So one microorganism, probably not. What I would do, I would probably introduce staphylococci at the levels that are not very high, and I will try to see, I will try to follow the changes at the molecular level to see if there is a trend in changes that may be considered as the trend leading towards the obesity. That's what I would do. But again, that's a question of a hen and egg. I think they're all connected. Thank you.
In one of the previous slides that Shikindas, we have seen that in the obese, the lactobacilli are increasing. The number of lactobacillus increase in obese, and in lean, the lactobacillus is decreasing. So that also sometimes put a question that the increase in probiotics are increasing the obesity or not.
And again, I think I marked this part of the table, and actually it was, I think, in a few tables, as something that really astonished me, and I couldn't answer these questions myself. And yet, when we force lactobacilli into the obese individual, we see positive effect. What should be preferred as high-fat diet for mice? They are described in the reference. If you will go to these slides, TOI numbers, all right, they'll give you direct link to the source of the paper, and you can find. But these are used by. It's a standardized diet. It's not something unique. There are standardized lean diets, fat diets, and they call this way fat-induced diet. They're even commercially available. I was kind of having more hopes to see a better effect with the bacteriocin producing probiotics, but vancomycin performed better. The hope is still with us, because we did see the changes towards lean profile of microbiota in these studies by the core.
But one thing is very clear, even some of these studies recently, that bacteriocin producing probiotics, they are able to modulate the gut microbiota. Even this kind of signal is positive for us, that these molecules are helping to modulate microbiota, and finally the phenotype will be affected ultimately. I will see the obesity is something which is a complex metabolic pathway. So a lot of things are involved in this, and control study and those things are required to specifically highlight that which one is active and which one is not. That requires several study letters, I think.
Right, and as I said before, it's really very difficult to, it's like when we're solving the mathematical equation, which contains multiple variables, what we want to do is to reduce the number of variables as much as possible, especially when it is possible to convert some of the variables into other ways of, presentation. And of course, the ideal situation is when we're solving the equation with just a single variable. All right. much the same, because when we are dealing with the biological systems, well, that's a kind of a message. Maybe it's wrong, but I stay with this message when I try to explain my students what we're doing when we're dealing with biology, and I'm saying that biology is the field of science where we're looking at chemical processes that obey the laws of physics, which can be explained using mathematical apparatus. All right, and if we cannot explain something now, it means that we do not have sufficient knowledge on some of the variables.
Yeah.
That's it. So mechanistic studies are extremely important, and I cannot emphasize it enough. It's not the science for the sake of the science. Let's do the things just because we're curious. And I think one of the famous people said in the past that being a scientist is a luxury. To be employed and to be paid to get a salary largely, being involved in the processes of satisfying your own curiosity, which may be true for fundamental science, for the pure fundamental science. But I think we are gathered here, we are equally interested in the fundamental science, in the applied science, and in the translational studies, which bring us, almost to the stage of the product. Okay, and fundamental science, of course, the knowledge, the curiosity, all the good things in us and humans, but also these should serve the purpose of understanding of how it works in order to make it beneficial for life in general.
Yeah.
More or less exercise with consuming probiotic can change the results of. Yes, it's not that I think that, I know that, because there were quite a few studies conducted where probiotic supplementation was given to the groups of individuals involved in exercising. And different groups were engaged in these kind of studies. There were those who were just exercising non-professionally as a part of their daily routine. There were those who were athletes at different level of professional involvement, and there were also athletes of the elite group of athletes. And these were separate studies. I want to emphasize that these were not studies where all these three groups were present. And there were studies done on mice who were forced to exercise, like to swim, for instance, right? And swimming mice is a very good, very potent type of experimental approach to see all kinds of things, their performance, in their energy, accumulated energy that is released towards, certain tasks, etc. So I'm very much for these kind of experiments. I hope that mice are not really suffering much when they're forced to swim. And in these kind of experiments, with probiotics involved, it has been shown that the exercise together with the probiotic can modulate the gut microbiota, number one. Number two, exercise when it is applied and when the probiotics are used, supplemented, probiotics can also decrease the effect of the stress. And what is interesting with the studies in humans is that the human subjects or professional athletes were studied during the exercise period prior to during the performance competitions and after the competition. So you can see different level of stress, different type of behavioral scenarios. And again, in these kind of comparative studies, it has been shown that the probiotic microorganisms can modulate gut microbiota. And of course, these studies were allowed because these are drugs and they're not influencing badly, the eukaryotic host and all of that. Some studies are done by the groups in New York. Some studies were done by the groups in England, Napier, sorry, not England, Scotland. Napier University in Edinburgh did a lot of studies which were very interesting. And they even have, I personally visited them, beautiful exercise facility, and several teams of professional athletes are volunteering to be involved in these kind of studies. And I think this is a very good example. You may consider doing these kind of studies at your locations. Similar studies were done with the groups of individuals who are high professional risk people. We're talking about the firefighters. We're talking about the law enforcement personnel. And again, these were separate studies in several countries, some in Brazil, some in Belarus, some in Israel. And again, there was some positive effect. And the interesting thing is that similar studies were also army personnel. About 20 years ago, these studies were performed in the US, so there was no positive effect at all, no effect whatsoever. And now, 20 years later, we see the studies with a positive effect. And my guess would be that the approach was different. The microorganisms used were different. And we can see positive results right now. So, short answer to the question, yes, I think that we can see better results. And the probiotics are not only helping together with the exercise to move towards the proper metabolism, right healthy metabolism, and to see the decline in obesity, but also help to improve performance of individuals who are professionally at the highest stress level because of the things that are involved.
Dr. Chikinda, there is a question comes with probably this student is working with the high fat diet in the mice. So she is asking what should be preferred as high fat diet in the mice.
Oh, I already answered it. I already answered this question. That was the first question. Yeah. That was the first question. I already answered the first question. All right. So we have 5 minutes break, which will I will need to change the slides, and in 5 minutes the break for everybody else, and we will go through another topic, which is functional genomics, and engineering of bacteriaceae. Well, obviously, I'm always my tongue is slipping to bacteriaceae because that's what I work on and that's what I love most. But yeah, probiotics, of course.
Yes.
My tongue is slipping, and it's like, of course I don't have an excuse, but it's still early hour for me. Was the Yakult trip yesterday?
Oh, we had today. For you, it is yesterday.
You've had the trip today, the Yakult trip today?
Today we had at 12:30 in the noon.
Okay, I'm not advertising the company, and I'm not involved with the company in any professional way or paid way, but I want to raise my personal, bottle, and that's my personal Yakult supplement for every morning. And I don't know if it helps me. I like the taste, and I believe in this product.
Oh, I like to. It seems tasty. picture, I think.
Yes, we can. Let me start sharing with you. Share. All right. Oh my goodness, functional genomics and engineering. I overestimated what I want to squeeze into the short period of 1 hour. I still decided not to remove any slides. And probably I will have to rush through some of them. The reason I decided not to remove them is one and only one. I wanted to share this information with the group. I wanted to give the group at least some idea. I know that the CRISP technology is a very fancy thing, which is now studied by many. Not really studied. Let's face it, it's a tool. It's a very good, very elegant tool that is used to design new microorganisms with new functions. I honestly believe that this tool may lead us to the formation of a good cassette of probiotics for medicinal targeted applications. I still believe that this tool is far from being useful for the over-the-counter and food applications, because these are the areas where the consumers are still hesitant to take the engineered microorganisms. Although we do know that some of them are engineered, we know that Danone is using engineered probiotics in their products, and they're just writing biotechnologically modified or whatever. We do know that they are. I did say a word about the laboratory evolution. I'm personally very much in favor of laboratory evolution. I just want you to know about it, some kind of check mark in your brains. Look for the laboratory evolution as the powerful tool, which is also currently employed in these kind of studies. And I hope I will have time, and again I will probably have to rush through the things related to some of the studies that we did in our laboratories here at Rutgers. And so obviously we'll start with the outline of our journey. We'll introduce ourselves into the microbes, how we depend on them and what they do with them, turn to E. coli, which amazingly, surprisingly at all for all of us, are both pathogens and probiotics, one family in one jar. We will then move to the functional genomics of lactic acid bacteria, then to probiogenomics back to 1917, and then we'll have the conclusion remarks, and back to 1917 is the work that we have done here in collaboration with several labs at Rutgers. Starting with the introduction, and so many new things, so many new definitions, and often I'm trying to be politically correct, but often when I see these definitions, I see the old things, the old body in the new clothes, and the fancy definitions. I really don't know what kind of role they're playing. It's like when I'm talking about nanotechnology, nanoformulations, and we'll be talking about it in one more session. Probably I will again repeat myself. I'm saying it's just the colloidal chemistry. So anyway, some of the things we need to introduce: genotyping, identification of differences in DNA sequences, genotypes, epigenetics, study of how biochemical modifications or interactions of DNA and chromatin when we're dealing with the eukaryotic influence regulation of the genes in the cells, and meta-analysis, combination of data from the experiments which were already published aimed at the creation of these statistically powerful models of the processes, which should lead to better understanding of the relations at the variety of levels, molecular level, organ tissue level, the entire organism level, but especially important when it comes to the seemingly small changes, subtle events. And I mentioned it before that, on the lower taxonomical level, at the level, for instance, of species, some microorganisms, even present in the small numbers, can play more significant roles. So this is the hope for metagenetic analysis. Functional genomics, study of how genes and the nutrigenetic regions of the genome contribute to different biological processes. And in particular, studying genes or regions on a genome-wide scale. So we're talking about multiple genes, multiple regions that work at the same time. I cannot resist from referring back to obesity. And we didn't say the word, genetic level, we didn't say these words, but we've had it in mind when we were talking about the combination of factors which are interconnected. And as a result of their combined action, we see one or another type of phenotypic changes or health changes in the eukaryotic host. The goal is to determine how these separate individual components work together to produce a phenotype. Again, back to obesity. Functional genomics main target dynamic expression of gene products in a specific context. So we have the obese phenotype, which genes are overexpressed, which genes are inhibited. We have the introduction of the microorganisms, which genes are expressed, which genes are inhibited. We have the microorganism that is a probiotic, suddenly starts to behave as a pathogen, which genes are suppressed, which genes are expressed more. We have knowledge, but these are the things that functional genomics should be addressing. And of course, it's not coming from nowhere. It uses the foundation of our pre-existing knowledge of the gene function relation. Functional genomic approaches, there are several levels. We will repeat it again and again through the course of this session. DNA, RNA, protein, and metabolite. Why? Presence of the genes is important, but will these genes really be expressed at RNA level? If these genes are expressed, will these RNA be translated into the protein, which may or may not happen, because we know that the regulation at the RNA level also takes place. We can have enormous amount of RNA, but it can be also destroyed, and the protein will never be produced. The protein is produced, it's there, but do we have the metabolic products, which may be real functional products that we want to trace to the phenotype? So DNA, RNA, protein, and the metabolic products. I said it before, I will repeat it again, that we should give very high opinion of ourselves. So you may think that we're like, the highest possible creation in the evolution. But in reality, we're a combination of ecological niches readily available for the microorganisms to populate. And, if we look, for instance, at the path of the lactic acid bacteria, of course, lactic acid bacteria, because we're dealing with them, in all our sessions and all our work, millions of years is, originally lactobacilli were actually associated more with plants than with animals, than with humans. And now, just thousands of years, we have the association with humans through the fermented products, so even closer association. And if you look at these kind of things at the evolutionary scale, it's really happening just recently, right? The table on the left, we have the number of microbial species in different eukaryotic organisms. What is the take-home message here? In the plant seeds and roots, they're not, all organisms. We have relatively small number. Drosophila melanica, the fruit fly, just 74. Medicine, just 40. All right, human got more than 1,000. Right, ape got almost 9,000. Why? Why do they need such a diversity? I don't know. Pig got still, significant number, but much smaller than in the humans. And we do know that, many processes taken in humans and in the pigs carry some similarities, for example, more in some cases than with the primates. So, take home message. In some cases, just a few microorganisms is enough to give the full capacity of the normal function of the eukaryotic host. And it doesn't mean that this is a primitive host. Not at all. We're not more primitive than the apes, I hope. But we have, smaller number of the microorganisms in our gut, And the pigs, I wouldn't say that these animals are not smart, but they have significantly smaller number of these species in their gut as compared to humans. What is the number that is sufficient to deliver the function? I don't have this answer to these questions. We'll see some examples, even with the probiotic bacteria, when the use of probiotics is causing the decline in the microbial diversity. And we still don't know whether it is okay and whether these eukaryotic host is still to be considered as fully functional and healthy. Mode of transmission and this is where we're talking about symbiotic relation, where there is a relation between the microorganism and the host. The cow rumen microbiota, the whale, and the human, and the reason we have three of them is because of very different environments. The ruminant eukaryotes have a very different digestion system. The microorganisms involved are very different. Whales, it's the aquatic environment, human and mice, of course, it's a different story altogether. So physical contact with the parents, physical contact with the mother, and we have the advantage from the birth canal contact from the environment, of course, as well. The environment here as well. With the whales, it's as open environment as possible because the water is being passed through the body constantly. All right. All the nutrients are largely coming from cellulose when we're dealing with the cows. In regard to the whales, heating and other complex organics, and a variety of nutritional sources for humans and for the mice, and especially for those that are in captivity in the labs and at home. So we have an understanding of holobiont, which is a central organism like a human being, and that includes the consortium of microorganisms that are engaged in all the processes that are happening in the eukaryotic host. Fraction of LAB in the gut is low, barely 1% in only a few persons. Take home message, they are few, but they are extremely, right? So we see vaginal environment estimated LB fraction less than 10 to the negative 1, total number 10 to the 6, 10 to the 7. Yet when we were talking about vaginal health, we're talking about the incredibly important role of lactobacilli. Oral cavity. Strep salivarius is perhaps the most important as a probiotic candidate and as a probiotic bacteria. Low numbers of LAB, but oral cavity is the first place where they're coming when they're being passed through the gastrointestinal tract. So this is an important location for the body. And then again, on the skin, 10 to the negative 2 compared to 10 to the 4 to the 7 of other bacteria. A large intestine, less than 10 to the 3 compared to 10 to the 9, 10 to the 11, and yet again a very function. So take home message, don't judge by the numbers, judge by the function. The human GI tract contains of microbiota diversity is beyond our imagination, the number of microorganisms. And I'm not even talking here about the viable but not culturable or non-culturable microorganisms. And we know that there are many of them. And of course, once again, when they're fancy words, simple message, I told you that these are fancy words. Which microbes are there? These can be found through the nucleic analyses, and these may help us to identify also viable but not culturable, non-culturable, because we cannot culture them using the methods that are currently at hand. What are they doing? Are they protein the products level studies involved, and what is the genetic potential, the capacity, what is sitting silently that we can utilize for our benefit, or what is sitting silently and can turn nasty or can turn good. Obviously, DNA and metagenomic analysis should give us answer in this area. So, I already mentioned uncultured microbes and the domination of Firmicutes, Bacteroides, and Archaeobacteria. Firmicutes and Bacteroides were mentioned in our previous session, and the healthy human subject, from one to another, the microbiota is not exactly the same. It's changing. It depends on many factors, and it's considered to be less stable and with a low diversity in infants, and then through the maturation of the individual, it stabilizes and comes to the condition where we have a diversity, appropriate diversity of the microorganisms in the young active adults through the entire life, then the decline obviously with the aging. Turning to E. coli and talking about these creatures, which can be both pathogens and probiotics. And strikingly enough, they can even share some of the parameters. I want to start with Niesler 1917. And 1917 is when these microorganisms was discovered. I give you the picture, the photograph of Dr. Niesler. And it happened during the World War I, and the entire platoon in the German army had a diary except for one soldier. And Dr. Niesler, being a good physician and a real scientist, he decided to take a look what is different with this soldier who didn't develop diary. And this is how these microorganisms, Niesler 1917, was isolated. So we can say, I guess with some level of confidence, that Niesler 1917 is perhaps the first probiotic known, and the first microorganism, I think one of the first at least, that started to be produced commercially as a probiotic bacteria. So what we have here is a Venn diagram with numbers of significant up and down regulated genes in several microorganisms. So we have here the we have Niesler 1917, which is healthy, which is a probiotic bacteria. We have CFK073, which is the urine pathogenic E. coli UPAC. We also have the ancestor organism, 83972, which is the ancestor to both of them. And we have different conditions of growth and different levels of the gene expressions in all of them. And we have the MOPS, which is the artificial medium with UV. We have MOPS with the formulation that promotes biofilm formation, and we have urine biofilm. So it's kind of, all three variations are presented here. And not surprisingly to me, we have, significant overlap, and that's what we see in the middle, in the up and down regulated genes. And when I'm talking about, 10 or 20 or 100, we have hundreds of genes. And take-home message is that these 97 express many genes that are associated with the uropathogenic E. coli. And these all three strains are closely related. So you may remember when we were talking about the selection of the microorganisms, I said that I am hesitant to accept the suggestion to consider some species as probiotic. I still firmly believe that the pathogenicity, unless we're talking about, for instance, salmonella, I don't know, pathogenicity and health promoting activities should be identified and confirmed on the strain, at the strain level. If we look again at the correlations in the matrices of expression of certain functions, again, it's 27, it's a very comprehensive 27 microarrays. And then again, we have Nislin-917, which is a probiotic. We have CFDO73, which is the pathogen. We have the ancestral strain 83-1972, and the combination of different conditions, the synthetic media, the media which promotes biofilm formation, and the urine. And again, we see the take-home messages that two urinary tract infection isolates do not cluster together. Before they are clustering with Nislin, so they are more closely related to the probiotic bacteria than to each other, which again may come to some as a surprise. So how can we interpret these data? Well, F1C fimbriae play a role in biofilm formation, they play a role in the mobility. And you take some literature and you see that, mobility is the pathogenicity associated phenotype factor. Ability to form biofilm is a pathogenicity associated factor. And in some papers, it's written in a very bold manner. And I cannot accept it because, good bacteria should also float, should also swim, should also go one way or another, should be able to form biofilm if they can't do that. So it depends. The correct answer on whether there is a pathogenicity feature or not, it depends. It depends on what kind of microorganism you're dealing with. And so the expression of F1C fimbriae doesn't play an important role during the late stages of biofiltration in the urine, but it does play an important role in the initial stages. Because once the biofilm is formed, who cares? All right, until the cells will start leaving the biofilm, and then of course these genes will have to be activated again. All right, so what we see here is similarities, but we also see the action of the genes and their products through the lifespan of the microorganism and through the lifespan not of just single microorganism, but also the microbial community. Why is that important? Because we're also looking at the microorganisms in relation to their eukaryotic host through the lifespan of the eukaryotic organism in different conditions. Functional genomics in LAD. So it's used to understand the response of the probiotic organisms, self-promoting bacteria, to their environment. It requires analysis of the genes involved in colonization, persistence, interaction, signaling, etc., etc. Colonization and persistence, of course, yes, only in the case when we're dealing with the colonizers, either transient or real colonizers. Signaling, by all means, yes, because this is one of the major functions of the probiotic microorganisms, interaction with the host through modulation of the signals to the three components: immune system, endocrine system, and the central nervous system. And of course, paleogenome resequencing will provide opportunities in apply genome, especially in the characterization of the non-GMO strains. GMO strains is a different story altogether, because we will have the specific changes introduced artificially, and we know what we're looking for. With the wild type organisms, we really have to study what they have to offer us. All right? And if we will look at the phylogenetic tree of, based on the sequence of several, in this case, 7 housekeeping genes from 36 different lactic acid bacteria, we understand, recognize the versatility of a variety of species. And we can see here we have Lactobacilli, Druki, Fermentum, Acidophilus, Johnsoni, Gasseri. So some of them Helveticus, Inneris, some of them like Bulgaricus, obviously in the fermented dairy products, as well as Acidophilus. Some of them as Crispatus and Inneris are obviously, and Gasseri are in the vaginal environment. It's very important for the health there. All right, so take-home message: versatility, the ecological versatility, but doesn't further detail its ecological role. So the important thing is, what are they doing on the spot, transient or endogenous involvement in the scenarios in the eukaryotic host. We see the diversity, we see that they're smart, we see that they're clever, we see how they interact, how they're related to each other, but we need additional things to understand what they do and how they do. Who are they? Or you mentioned that they're less than 1% in gastrointestinal tract. And some may even think that they're not endogenous, they're just passive, they're just transit, they're just using this train to hold human body or any other eukaryotic organism to pass through the time. Some LAB, however, have high interspecies diversity, and they include both endogenous and passenger strains. And that is especially clear when we study the non-colonizing Lactococcus lactis, and we should mention that there are a few papers studying Lactococcus lactis as probiotic bacteria, but not really that many. They're non-colonizers. Adaptability, biological tolerance, specific host adaptation, resistance to low pH, etc., etc. So these are the factors that are engaged. Prevalence in oral cavity, again, not that high numbers, but I can't emphasize enough the importance of them. Although I was putting major emphasis of Strepsillivarius because of the probiotics that belong to this group. Vaginal environment, we're talking rather old Crispatus, Gasseri. Inneris is underlined for me not to forget to say that yes, this is a major organism, but it can have 2 roles. It can be healthy, but it can also be pathogenic. But how this switch goes, we still don't know, and this is still a kind of a gray area. Comparative genomic analyses of lactic acid bacillus. This is a very interesting experiment where we have several microorganisms studied, and we have LGG, and they're kind of related if you look at the B, which is a comparison of relevant stress response genes, or in L. rhamnosus, and they're quite similar, right? Except for some things, we have about 90-something% identity 96%, and then 98. And in one strain, I want to bring your attention to this MC143, there is a deletion, so the function is definitely disrupted. So the origin, some of them are from the fermented products, one of them is MCO10 from the infant stool, LGG obviously is our beloved probiotic, MC143 is from the infant stool, and there is a strain with a deletion, right? And this is definitely where the function is disrupted. And when we look at the biosalt hydrolase, yes, the deletion. When we look at the situation with the survivability after the exposure to the oxygal bile, and again, what we see here is a significant difference, especially when we compare the strain with the deletion of the strain, which is LGG strain, and the transcription analysis shows really very interesting changes. MC143 have really very low level, and that is where the deletion is seen. All right, so definitely the entire path from genetic level to the RNA level to the level of the gene product and to the level of the strain behavior in different conditions. We can see similarities, but we can see differences in the microorganism. So what are the take-home messages from the slide that we saw before? Different profiles, and in each of the strains we have unique regulation system. In vitro physiological analysis allows to mimic gut environment, and this is where we have, for instance, the exposure to the low pH, the exposure to the bile salt, and we can see the situations, and I mentioned and pointed to it, that the growth of the strain with the deletion was inhibited, but for instance, LGG was performing really well. Strangely enough, 143 strain with a deletion survived better than other strains after exposure to the bile. It didn't grow well, but it survived better. And we see that, for instance, the reverse transcription qPCR targeting the bile salt hydrolase gene showed us that this gene was expressed poorly. So what are the factors, the other factors that allow these microorganisms to survive better? And we also have the outcome of experiments where the CPSH gene, the hydrolase gene, was disrupted through the insertion, and we have an assumption to make that it would be detrimental to the survival during the bile stress. And final take-home message: findings show that coupling of classical microbiology with functional genomic methods for the characterization of material strains is critical for the development of novel probiotics, as variability between strains can dramatically alter material physiology and functionality. Translated into the plain message, multiple functions can be delivered by various genes, gene products, and we can see that, especially with this example, that survivability in the given condition may not necessarily be caused by the activity of a certain gene that is involved in this process in other probiotic bacteria. How we functionally characterize probiotics, and again, what a surprise, we have proteomics, functional genomics, and transcriptomics, where we deal with the characterization of proteins expressed by the health-promoting microorganisms. The gene function study, which allows us also to utilize mutations such as the hydrolase mutation that was used in the previously described study. And of course, DNA, microarrays, and RNA sequencing, RT-qPCR analysis can also give us some information, shed a light on what is happening with the expression of one or another type of genes, group of genes, under certain given conditions in the environment that the probiotic microorganism has to face. And with these, when changing page, going to another fancy term, probiogenomics. How probiogenomics are different from other genomics, I don't think they're different in any way except for one. This is the genomics that are focused, concentrated on health-promoting microorganisms. So it's a tool to obtain genetic insight into adaptation of probiotic bacteria in the human. Otherwise, it is the same. And of course, how do we select the microorganisms, the probiotic microorganisms, using this omics approach? And you may remember this picture from our first day gathering, right? And on the right, there was a simplified, more kind of, in quotes, reasonable flow chart. And I'm still asking, is that really very different from the traditional approach? I don't think so. It has more insight, it has more specific details, but as a flow chart, as a strategy, it's still the same. We isolate the microorganisms, we identify the conditions under which it's culturable, we look at the safety of these organisms, we look at the specific functions, and as previously I said, that safety must come first. If it is not safe, who cares? We look at functional aspects, we look at the health-promoting aspects, and we look at the technological, whether these organisms can be produced at a large scale. It's the same, it's not different. It's just, closer zoom with the specifics in what needs to be studied. Probiogenomic analysis, and again, fancy words for simple things. All right, enterococcus safety property. Why is that? You may remember I mentioned that enterococcus can be both good and bad. And enterococcus are involved, for instance, in fermentation of cheeses, of many products, fermented dairy products, but at the same time they can be pathogenic. So virulence genes, antibiotic resistance are very important. Molecular signatures revealed peeling, subunit, endocarditis, antigen, cytolysin, yes, by all means, but is it genomic, whatever, probiogenomic analysis? Just a genetic analysis, to be honest with you. Metabolomics, biogenic amines, no significant tyramine, putrescine, cadaverine, histamine, spermidine, which is not a surprise, to be honest. LA5, lactobacillus is very well studied, as well as BP12. These are in the group, in the category of most studied probiotic bacteria. Virulence in L. helveticus, hemolysins, but some hemolysins are revealed, or some hemolytic in lactobacillus. This is not really something different, something unknown. Antibiotic resistance, yes, it is very important. Plus AB lactamase, penicillin-binding protein, etc. And again, you may remember we're talking about it. The most important thing for the antibiotic-resistant marker is not to be located on the mobile elements. This is the take-home message. On the other hand, the antibiotic resistance may be useful for some probiotics, especially when we're dealing with a scenario where the antibiotic and probiotic treatments are coming together, or sequential use of probiotic after the antibiotic is implemented. All right. Safety property bacterial production, I'm not sure how that may be a safety-related issue. It's a rather good thing. A class 2 bacteriocins not described, maybe other bacteriocins can be revealed, who knows. Animal studies and associated probiogenomic analysis. right? So the strains Streptococci, bacilli, finally my beloved spore formers, Plantarum, Casei, Bifidobacteria, health claim or disease obesity, diarrhea, control of diarrhea, suppression of diarrhea for bacilli, lactobacilli, etc. Of course, this is animal studies. The outcomes we see the decline in body weight, smaller water excretion, of course, with the diarrhea cases. Tissue integrity is improved with the improvement of the mucous barrier declination. I'm curious, I mean, these kind of things should be observed also with bacilli sublet, but they didn't look at metabolomics, membrane vesicle characteristics, transcriptomics, the expression level of the biomarkers, metabolomics, characterization of the cellular components, and proteomics, lipoproteins characterization, and all these characterizations. I mean, they're important, but they're not that crucial for the actual outcomes they help to understand, and they should certainly come together with the genetic analysis. Human studies, similar things with the human studies, and again we have IBS, insulin resistance management, lactose intolerance. I wouldn't pay much attention to these nowadays because, as I mentioned before, lactose-free products are already on the market, and the number and variety is increasing. Obesity management really very important. Bifidobacterial health, randomized double-blind placebo-controlled trial, really very good age distribution from the younger active adults to the mature adult and elderly declining population. Body fat mass decline, which is a very good result, and the genomics, metabolomics, we have the fat and metabolic production for the beneficial for these condition improvement constituents studied. So good results overall, and we have the molecular level of understanding. And again, for instance, insulin resistance, right? Let's take a look at this. So the improved glucose metabolism and on the genomics level, carbohydrate metabolism pathways were studied and correlated to the improved condition, which is again a good result. When we look at the potential future trends in probiotic genomic fields, we obviously start with the candidate strains, bearing in mind that there are applications in the food, feed, and pharmaceutical areas. So we compare the isolates, we see what their potential through this fancy probiotic genomics. We understand what they can do, what is their potential that is already residing as a capacity of the cell, which can be improved either through improvement of the existing things or can be improved by introducing new genes. That is bioengineered probiotics, which then go through the preclinical studies, the regulations, and finally, hopefully, improved for the use in medicinal applications, agriculture, and human health. And with that, we're turning the page and going to walk through the CRISP system. And with these, I will go with you through the schematics several times, giving, seemingly different, but pretty much the same pictures, different angle of the same things, what can be done there and how it can be done, and giving examples also in few slides, I think, with different types of health-promoting bacteria, lactobacilli, bacilli, I think yeast, with some examples, specific examples of what can be done using these kind of approach. And this is basically the gene editing approach. It consists of two components. It's a guide RNA to measure the target and the CRISPR-associated protein A, which is an endonuclease. It causes double-stranded DNA disruption, the break. allowing the modification to the genome. You have different color codings, which are explained here, and the vocabulary for the acronyms used in these pictures. So we have the double strand break, and then we can have either homology direct repair, or we have a non-homology end joining, which allow for instance, random insertions in a variety of locations, which may lead to the functions that are desired. And because it is a directed approach in changing of the genome, we have an ability to create what we really want to see. So on this note, again, the schematics in a bullet form, if you wish, we have the clustered regularly interspace palindromic repeats, which is the crease, and they are the sequences in the bacterial genome, and they deliver protection against a variety of stressors such as viruses, and when combined with the crease-associated proteins. So Cas9, I already mentioned it before, is an endonuclease, and it cuts both strengths of the DNA molecule, and is directed to the target by section of RNA. And these can be synthesized as a single-strand cholestatic single-stranded RNA, sgRNA. And the section of RNA that binds to the genomic DNA is in the size of 18 to 20 nucleotides. So in order to cut the DNA sequence, like 2 to 5 nucleotides must lie at the 3' of the guide RNA. And this is called the protospacer adjusted molecule. All right, these are again the vocabulary that we need to keep in mind when we're dealing with the CRISPR system. Cutting, what's next? Repair. And we need to have repair, and I already mentioned in the picture you saw on the left was the homological repair, on the right there was non-homological repair, non-homology, and joining typically leading to the random insertion or deletion of the DNA, and the homology-directed repair is happening when we have a homologous piece of DNA, obviously, used for the repair of the disrupted molecule. Most important take-home message, allows for the precise genome editing. So we're introducing the changes exactly as we want to see in one or another time of the locations and the genome. All right. So we're moving, this is a kind of a comprehensive flow chart, and I personally like it, and to make it easier to follow, because where is the A, where is C, where is D, I put the arrows here so we can go through them. So some of the examples of applications in the engineering, we're starting with the A. We have the Cas9 mediated editing, that relies on the obviously the Cas9 function, including targeted DNA molecule, and the protospace adjustment motive, the PAM motive, allows for the Cas mediated recognition and cleavage. Then we have the double picking, that's a P part, which paired with the Cas9 nucleus for the following engineering process, and that's the part B, where we have double nicking with parent CNS nuclease for this kind of engineering. Then we go to part C, and this is where we have the catalytically inactive Cas9 variants, which is Cas9, and that's in part B. Transcription regulation occurs in maybe CRISPR activation or CRISPR interference, and the transcription regulation goes under the use of the Cas9, which is a mutant form of Cas9, and it can bind but not cleave the DNA molecule. And Cas9 controls the expression by inhibiting transcription. You can see activator and repressor here that are engaged in the process. All right, the key is the CRISPR-based visualization, so we're taking kind of a closer look at it, and finally we're moving to the final part, negative selection. CRISPR negative selection, where Cas9 can also be utilized to cleave the unmodified DNA, because you need to get rid of it. And all right, so this way you are conducting the negative selection of whatever you want to achieve. And I promise there will be several examples. So we have pedicocccus acidolactis A, we have in the B, which is below B subtilis editing. C is actually yeast, and we do know that Saccharomyces cerevisiae are working as probiotic bacteria. And finally, last but not least, D is Bifidobacterium, which is also a known probiotic. So in the A fraction, we have the objective. The objective was to enhance the production of lactic acid. This is very important. Also technologically, not necessarily probiotics. For instance, if you want to produce the fermented bread where lactic acid bacteria are heavily involved in fermentation, providing acidities through lower pH and providing the flavor at the same time, and to make it biologically acceptable, you need to improve the lactic acid production, shorten the time of the fermentation without damaging the flavor and other characteristics that are coming from this lactic acid involved in the process. And well, you can basically include the method, the CRISPR method, to improve lactic acid production without jeopardizing other functions. All right. B is the editing in B subtilis with the improvement with the synthesis of N-acetylglucosamine and acetylene. Now, I do not know exactly why these, some of the possible needs. Acetylene, for instance, is also involved in the flavor production in some fermented food products. So, as an example, it's a good example showing that the specific functions can be introduced into the microorganism without jeopardizing the rest of the genome. Bifidobacterium deal, and this is where the CRISPR approach was used for large DNA fragment knockout and introduction of the point mutations in the genome of Bifidobacterium. Strategies for probiotic microorganisms. What we have here with the important components that must be considered: vectors, screening markers, and of course drug resistance can be used in the initial steps, but not at the later steps where you want to have the probiotic microorganism with no genes coding for antibiotic resistance. Nucleases, DNA cleavage, of course, and this is where we have Cas9, 12, Cas9, Cas3, and many other of them. Transformation, whether you can also bring these microorganisms, engineered microorganisms, to the stage where the modified genetic elements can be transferred to another organism. These can be done in a variety of ways. Some microorganisms can be really useful for electroporation, chemical physical methods, combination, calcium-induced competence, conjugation can also be used. Protoplasm can be used as well for the transfer of the genes. And last but not least, of course, the regulation. Post-therapeutic application of engineered probiotics based on CRISPR system. And again, some of the examples include infectious diseases, IBS, obesity, all the things that we were talking about before. So in the case of infectious diseases, engineered E. coli delivers CRISPR-Cas9 in gut and eliminates antibiotic-resistant bacteria, which is a very good take-home message. In IBS, we have engineered Saccharomyces expression with the human pyridinogen receptor and secreting adenosine triphosphate degrading enzyme, and that can sense and degrade the the pro-inflammatory molecules in the environment. And finally, last but not least, the obesity, and we have engineered bacillus subtilis, which produces butyrate and can also assist with the regulation of metabolism to prevent obesity. And uric acid is really very important in a player in that. Probiotic guided CAR T cell for solid tumor targeting, that's another example. And this is where we have targeting therapies such as chimeric antigen receptor cells, which is a very potent tool in some degree, can selectively colonize privileged tumor cores and can be engineered as a carrier of the vehicles directed towards the target, the tumor target. And so the probiotic guided CAR T cells, or CAR-F cells, were developed. The tumor colonizing, color-promoting microorganisms, they can release the synthetic targets that can also identify and label tumor tissue for CAR-mediated lysis. And this system demonstrated the good potential. Take-home message: yes, multifunctional probiotics can be engineered to release hemateins to improve the CAR T cell recruitment and the overall therapeutic efficacy of the method. Kind of a closing to the end of what we want to talk about here, and then I'm moving to an example. I hope I will have time, and if not, maybe 5 minutes I will steal from your time to finish with today's gathering. So what we have here is synthetic CAR targets, tags, which produced and released in situ by tumor colonizing probiotic. And again, we have Isla 1917. And Isla 1917 is used as a very potent platform for engineering of a variety of probiotics with a specific target action. All right, and they can be labeled with the components of solid tumor or de novo lysis of GFP CARs. And tags are designed as dimers of SFGFP fused to the HPD that bind to cell surface and matrix proteins found in the TME, which is necrotic tumor microenvironments. And this is how they work in the schematic representation. So, multifunctional probiotics, they produce combination of chemomodulating factors, which may facilitate systemic delivery and delay the growth of orthotopic breast tumors. That's another example. And this is where probiotics are engineered to release and activate mutant of the human hematin to recruit CXCR6 or CAR T cells directly to the tumor site. And the treatment of subcutaneous TNBC tumor with the pro-combo strain, followed by the intravenous from CAR T cell reduced tumor, and that was done in the rodents and without affecting of the body weight of the experimental mice. We're changing the gears, going back to this lab. This is where I want to introduce you to some experiments done on the engineering. Again, ISO 1970, which for the production of beta-carotene, which is a precursor of vitamin A. Please pay attention to the right figure. This study was published in the Journal of Nutrition. The Journal of Nutrition allows actually that came on the front page of the journal, and it took a while to make the mouse to line over the bowl with yogurt and the carrots. And take a look at this picture. They don't allow photoshopping. It's prohibited by the journal. So it took a while to catch the mouse that is living on the bowl. But something was not original in this picture. I'm just looking at the audience to see how observant the audience is and what is not real in this picture. Anyway, mice, bacteria, and beta-carotene. I bought these little cute mice. I still have one of them hanging from the shelf here, and I gave it as a present to all the participants of the team. Really color, somewhat similar to what we see in GIAN. This was actually sponsored by Bill and Melinda Gates Foundation. And yeah, just briefly to mention that vitamin A is a very important component of nutrition, and if there is a lack of vitamin A, there can be blindness, there can be decline in the immune system causing the severe infections, even risk of mortality. And vitamin A supplementations, yes, they exist, and different ways of delivery of vitamin A are also known, and you can use vitamin A supplements. You can also fortify the delivery of vitamin A, but then this stability becomes an issue. You can diversify with the access through a variety of foods, but then will these food be acceptable by the consumers is a big question, of course. Biofortification is another approach, but breeding time is really very long, and again, cultural acceptance is an issue. Alternative strategies, what are they? During convention, beta-carotene, which is a precursor to vitamin A, and there were some attempts for microbiological production of beta-carotene many years ago. Nobody really tried to engineer bacteria to rescue vitamin A deficiency, for instance, in animals, and that is why we decided to do that. And many microorganisms can produce, the beta-carotene naturally, such as like different fungi, different yeasts like Saccharomyces or microalgae. And there are a few bacterial species like Capricola, Serratia marcescens, that can also produce beta-carotene at relatively low level. And the problem with these strains is that, in the first one can be the opportunistic pathogen, and another one, the level of beta-carotene production is relatively low. So we need to move away from the possibly pathogenic microorganism and to increase the production level. And so what we decided to do is first to start with E. coli kind 12, and then to go to MG1655. MG65 is the non-pathogenic microorganism, and the mutant MG65 with star was obtained by one research group, and this mutant has better ability to colonize the gut of the mice. And so we wanted to see if we can produce beta-carotene in these microorganisms to rescue the deficiency in the animals. And so, the microorganism was engineered, and it was delivered to mice. Two different types of delivery were used, single dose and multi-dose delivery options. Of course, it comes from not from nowhere. There are different regimens described in the literature, and so we followed these regimens. And what we see, and we look at, for instance, the concentration of feces of the microorganism of beta-carotene, and the last one is the mean concentration of MG1655 with the beta-carotene genes isolated from mice. And the bottom line, the take-home message, yes, MG1655 with the star can colonize, and we can see these microorganisms being shed in the feces, and we see some level, not high, but still some level of beta-carotene. Keywords, some level, but not high. So what needs to be done is to improve the production, and the beta-carotene levels were improved compared to the previous studies, but we want to take advantage of the plasmid. And this is a plasmid that carries two additional genes, the initial gene of the pathway and the final step of the pathway. And we made an assumption that adding these genes, the helper genes, can also assist with the improvement of the beta-carotene biosynthesis in the system. And what we see here is the picture, of course, and the numbers, of course, with the original construction where the genes, the entire pathway genes are present in a packed beta vector, and the same microorganism, but in addition, the helper plasmid was introduced with the first and the last genes. And I hope you can see with the eyeball the differences in the color, and of course we see the differences at least, you know, with these kind of experiments 3 times difference. Next step, obviously, to put it into NISA 1917. Before that, it was K12, and it was done. And you know, the microorganism utilized was E. coli NISA 1917, and again we see the difference in the intensity of the orange color and the differences in the numbers of the beta carotene production in the cells. Now we look at the production, and this is where we have aerobic respiration. I want to emphasize this is important. Okay, this is like a normal lab environment. All right, and what we see here is the production goes, and CFUs are plotted at the top and the production at the bottom, and pretty much when the cells plateau and we have both the recombinant strain, the single plasmid and the helper plasmid, but the production largely goes when the microorganisms are in the stationary phase. And this is what we observe with the experiment where, you know, the mice were fed with the NISA 1917, and we have the levels of the beta carotene in the serum, liver, mucosa, feces. Basically not feces, not really much difference. Some difference in the mucosa, in the liver, and in the serum. Whether these differences are biologically significant, I'm hesitant to say, but there are some differences and they were observed. But what is important to mention here is that we're dealing with the environment, the gut environment, where the geometry, the profile is so complex. We have anaerobic, microaerophilic, anaerobic conditions, and these conditions are changing over the time with the food uptake, with the rest and other stressors and other conditions. So we need to see how the production goes not only aerobically but also anaerobically. I'm sorry, this is where I forgot to put the asterisk. So of course it's an asterisk. It's the strain with the improved ability to colonize. And differently from the scenario that we show in aerobic conditions, the production starts at the logarithmic phase and persists to some degree through the stationary, and then declines with the decline in numbers of the population. So different levels of production, which is not a discouragement, combined with the production in anaerobic conditions, tells us that actually we can achieve some levels in the environment where conditions are changing from, you know, microaerophilic to anaerobic to aerobic, depending on whatever is happening with the eukaryotic host. We still can deliver the certain levels of beta carotene in this environment. And aerobic production, again, we see here that 20 hours and 44 hours, 68 days, and this is with E. coli MG 1655. We have the good production, just kind of zoom, if you wish, at the level where the cells are present in more or less stationary situation. Kind of a decline start happening from 20 hours to 44 to 68 hours. Take-home message: should we have continuous delivery? Should we not worry about it because the conditions are changing, aerobic versus anaerobic? Should we think about the microorganism that we can effectively colonize, because we didn't actually look at the efficiency of colonization here? Again, anaerobic production zooming into 68 hours condition. And we see here that over this period of time, we have more or less, well, the deviation is kind of high, but we still observe some level of production. And what is important here is that we're dealing with the condition where the production is controlled by the inducible promoter. So basically, we can induce the production with the assistance of ion adhesion. Oops, sorry. Changing the gears, coming to the kind of zooming into the scenario of old-fashioned, classical, traditional approach. What we can say here is that a variety of old-fashioned but good, solid approaches can be used when we're dealing with medicinal application. Strong promoter, absolutely. The number of essential genes, and you saw the example where we added first and the last genes, and we had improved levels of production with these helper genes, and colonizers versus non-colonizers. Of course, it depends on what and where you want to use. Mechanism of action of engineered probiotics, and again, they're numerous, and I don't want to take much of your time. We're almost at the end of our today's conversation. Modulation of the immune system. We were talking about the exclusion of pathogens, competitive exclusion through the production of antimicrobials, competition for the nutrients, competition for the site of attachment, sensing and disease diagnostic. Of course, this is a very new field which is rapidly developing, detection of disease biomarkers, which can be done also with the assistance of probiotic bacteria and triggering responses of the eukaryotic host through the interaction with the host immune system and endocrine and nervous system. And of course, modification of the host metabolism through the production of the enzymes. And with this, I want to finish our today's gathering. And I would like to thank you all for your attention and for the patience. Thank you very much. And I think we're ready to go through the questions if we have some, and I can see. Okay, which tool or software is better for construction of phylogenetic tree, and which is the best method to construct a phylogenetic tree? That is not to me, and I mentioned that this is not my cup of tea. Actually, when I did these kind of studies with the phylogenetic tree of the bacilli probiotics, Bacillus subtilis compared 1933 and Bacillus amyloides fascians, these were done by the service company. I didn't do this kind of studies. Can we modify natural probiotic bacteria and make it release such proteins which can neutralize toxins or harm? Bacteria with it can help us fight many HSTT and cause bacteria. Okay, this is a two-component question. The first part of the question is whether we can engineer probiotics which are active in detoxification. The answer is yes, and moreover, these kind of detoxifying microorganisms are known not only as engineered but also identified through the natural selection processes. These microorganisms are used in bioremediation, and I think this is a very important thing. We mentioned in our previous gatherings, I think yesterday, that one of the, and also today, one of the things that is not really in the area, two of the things that not really in the focus of our gatherings, plant probiotics and soil beneficial microorganisms. So bioremediation in the soils where all kind of chemical fertilizers were used in the past, which are now forbidden. These kind of bacteria are known. Many of them belong to Pseudomonas. Pseudomonas putida is one of the microorganisms. Some of the Pseudomonas aeruginosa, some sporophyllin bacilli. Some microorganisms were reported as actually reducing mycotoxics. And what they can do, they can actually capture mycotoxins that are present in the environment, and mycotoxins can be removed from the eukaryotic host with the shedding microorganisms. So to answer the first part of your question, yes, there are microorganisms that can remove toxins, can work as detoxifying agents, both naturally isolated, and based on these naturally isolated, the engineered microorganisms can be created. Second part of your question, can we colonize vagina with these kind of microorganisms? I don't recall seeing really papers with a solid evidence on good colonization which persists, but I must admit that I didn't follow it very thoroughly in the last years, because my focus changed from vaginal applications to animals of agricultural importance. I occasionally follow this area, but the answer is why not? Number one. Number two, your question bears the hope for a silver bullet. Your question bears the hope for something that will have multiple functions against multiple infectious agents. This is the ideal situation. This is a hope which, unfortunately, in most of the cases, is not working. So why not think about a cocktail of microorganisms that synergize in biofilm formation and have ability to deliver multiple functions to cover several important targeted STD microorganisms. And saying that, I give you an idea for the, research grant application, right, which involves the study of the biofilm formation by the complex microbial populations, their action, and many, many more things. And you can engineer them to prevent the biofilm formation by them. You can engineer them to prevent communication, worm sensing, etc. And I was talking about bacterial vaginosis, and again, I am proud, not being a medical doctor, but being among those who, from day one, were talking about BV as an infection, not a condition. Number one. Number two, I'm very happy to see that nowadays the medical community recognize that BV associated microorganisms can be present in male subjects as well, and in fact they can be present in significant numbers asymptomatically. So these microorganisms can be transferred through the sexual intercourse to female subjects and develop into the bacterial vaginosis with the pronounced symptoms. So I think, again, we're thinking broader, from the development of these probiotics to the development of the formulations for the personal hygiene. Usually when we talk about personal hygiene, we assume like feminine personal hygiene, human personal hygiene. All right. Thank you.
Dr. Chikindas, I will take a minute only.
Yeah.
What about this bioengineered probiotics, whether it is acceptable by the ethical committees to use it further or not?
Right. That's a good question, and I guess the answer will be relatively short. Not for the food applications, not for the over-the-counter applications, definitely for the medicinal applications under the control of medical professionals. We see these kind of examples. In clinical trials, I don't know much about the actual treatments, but I would assume that if clinical trials are being conducted and they're successful, maybe in the real clinical treatments as well. But I encourage all of you, if those of you who are interested, keywords, Google it. Now we we often when we respond to the questions that we don't have time or ability to expand, we say Google it. Okay, Google it, and the keywords are laboratory evolution. All right, the laboratory evolution is, I put it in quotes, an non-invasive method. Non-invasive in a sense that we do not use, like, scissors and glue, if you wish to take some genes off, take some sequences on, or break some sequences in. So we're not doing this kind of pure engineering. What we're doing with the laboratory evolution, we're speeding up the process of selection. So it's the classical selection in the 21st century. And I'm personally very much in favor. I find it very exciting, very productive, and delivering a lot of very interesting and promising results. So Google it, laboratory evolution.
Thank you. I hope there is no question and you answered some of them, all of them actually. Thank you very much. See you tomorrow. Have a nice day.
You too. Thank you.